control on how much activity you really have going one way or the other if you can layer the control through separate hierarchical levels. Questions? Yeah.
Do different tissues, right, do they have different effects of all of these? So, tissues, let's say, right, compared to like your muscle.
Yes, these things are different in different tissues for sure, depending on what the physiological role is. And we're really just focusing on liver versus muscle. And we'll see this will sort of hopefully come full circle. By the end of the lecture today, we will see like the real core of what's different in muscle versus liver. But you're right, in general, it has different. It's different in different tissues and to different extents too. Yeah.
Just double checking. This is for glucose neogenesis and not for the breakdown of
Glycogen.
Yeah, glycogen.
Yeah. Right now we're talking about gluconeogenesis versus glycolysis.
Okay.
We're not talking about the glycogen breakdown buildup yet. Okay. Although the same hormones also have an effect there for the same reasons, but just on different enzymes. So there's another little complication on this F26BP. It's also affected by a metabolite in the pentose phosphate pathway. Right? So when glucose is high, right, we have a lot of glucose. That means you also have a lot of X, we call it X5P, that's xylulose sugar. If you go back to your pentose phosphate pathway and you look down that pathway, you'll see that at the end of the oxidative part of it, we have this xylulose 5-phosphate compound, which is a 5-carbon sugar that results after one of the carbons of glucose went up as CO2. Remember that stuff? And the pentose phosphate pathway, recall, was the pathway that makes a lot of NADPH, right, for fatty acid synthesis, and also can result in a ribose 5-phosphate for nucleotide synthesis. So it's a different way to oxidize glucose that generates different metabolites for different purposes. Okay, so if there's a lot of X5P, then that functions as an allosteric activator of an enzyme called PP2A. This is the structure of PP2A. What PP2A does, so it's MECPP, there's another one called PP1. So there's a number of different phosphatases, and the textbook doesn't always do a great job of distinguishing them. In this case, the phosphatase is called PP2A. Any phosphatase is an enzyme that removes a phosphate group from something else. And when it does that, it either activates the activity of that protein it's working on, or it inhibits it. Because the protein it's working on wants the phosphate for activity, or wants to get rid of it for activity. So here, if we take a phosphate off of this PFK2 FBPase2 enzyme, we activate PFK2 and we inhibit FBPase2, and that's what this is. Right? So if we take a phosphate off here, right, that's basically what this is. It's a different phosphatase than the one that's mentioned here, but it has the same effect in terms of moving you this way. Okay? So if we take a phosphate off, we've just activated PFK2, and that means we activated glycolysis. Because if PFK2 is active, then we have a lot of this guy, and a lot of this guy means glycolysis goes forward. Right? So we activate that guy, we inhibit the reverse reaction, the gluconeogenesis reaction. Right? So that means this goes up, so glycolysis increases, gluconeogenesis goes down. Right? Acetyl-CoA and NADPH levels rise. Right? Acetyl-CoA levels rise because glycolysis got activated, and therefore from pyruvate to acetyl-CoA. NADPH levels rise because you've got a lot of X5P, which means that you've been running that oxidative pathway, which is generating a lot of NADPH. So acetyl-CoA is the precursor for fatty acid synthesis, as we'll learn next week. NADPH is the reductant for fatty acid synthesis, as we'll also learn next week. So what this essentially is doing is providing a dietary link, like a nutritional link, between eating a lot of high carbs and having the carbs be convertible into fat. Okay. So there's a way to take carbohydrate intake and in a net conversion sense, turn it into fat. So if nutritionists or diet advocates tell you, don't eat your carbs, there's all these crazy diets out there, right? Human nutrition is so complicated that it never boils down to a simple thing like this. And no matter what diet you advocate for, you can find some enzyme somewhere that will kind of advocate in the direction you want it to. Right? But it is nonetheless the case that it is possible at least to go from a lot of carbohydrate and make the net synthesis of fat. It's actually not possible to go the other way. You can't affect the net synthesis of sugar if you start with fats or lipids. Right? That's probably not something we'll get into in the class. We may get to a slide that touches on it, but you can go from carbs to fat. Yeah.
Just to clarify, so was X5P high when glucose is high because it's the end product of the?
Yeah.
Okay.
Yeah. Yeah. Just because there's a lot of flux through that pathway because there's so much glucose around. Other questions? Okay. So you start to see how all these pathways kind of tie together. So there's another level of control, of course, that has to do with transcription. Those of you who want to take 492, which I heartily recommend, will learn a lot more about transcription. But there are a whole variety of proteins that work in the nucleus to increase the synthesis of transcriptional rates for certain messenger RNAs. And in this case, the messenger RNAs that get increased are the ones that are encoding pyruvate kinase and fatty acid synthesis enzymes. So all the kinds of enzymes that can be made when there's a lot of energy around. So fatty acid synthesis enzymes, if you have if energy-rich state, you can synthesize fatty acids. Energy-rich state, you'd be breaking down glucose. It's a little bit counterintuitive, actually. Regardless, so glucose comes in and through pentose phosphate pathway makes cellulose 5-phosphate, which activates that PP2A protein that we saw on the last slide. Okay. So when it activates PP2A, that dephosphorylates a protein called ChREBP, which is a carbohydrate response element binding protein. Carbohydrate response element is a piece of DNA in the nucleus. Binding protein is a protein that binds that piece of DNA in the nucleus. Right? So here we are in the nucleus, and X5P can get in the nucleus, this dephosphorylated protein can get in the nucleus, PP2A can get in the nucleus. It's a little bit messy as to why not just have both dephosphorylations occur here, but the ultimate effect is that dephosphorylated response protein here is available to bind the gene, and this ChORE is the carbohydrate response element, which is a piece of DNA. Right? Just a little element, maybe it's 100 nucleotides of DNA or 20 nucleotides of DNA, something like that. So this protein binds it, another protein called MLX also binds it, and that stimulates the synthesis of messenger RNAs. Okay? So a lot of glucose in the cell comes in, and messenger RNAs get stimulated to be made. What gets stimulated to be made? Excess carbs getting metabolized, fatty acid synthesis enzymes turning them into fats. Right? Excess glucose coming in. This is pentose phosphate, but you can also imagine you're running a lot of glycolysis, so you're running your pyruvate kinase. So the way to look at it is in terms of, not so much in terms of energy charge, but just in terms of, a lot of glucose coming in. So a lot of glucose coming in, you can make a lot of ATP, but let's say that we're before the point where we have so much that we want to run the pathways the other way. Does this make any sense? Am I being confusing? Please ask.
That last
Yeah.
part.
I guess the confusion is, or at least for me, is like when does the